So now that we've moved on from a bit of our background data about genomics, bioinformatics, and databases, I want to take a look at another function of bioinformatics. So what we've looked at so far, remember, is bioinformatics and public health when we did a blast search to determine nucleotide differences that were correlated with disease-causing genes. We took a look last week at bioinformatics and biodiversity and the Barcode of Life database, where we can determine the identity of an already barcoded organism through a simple blast search. And it also introduced the idea of determining or picking a gene for the Barcode of Life database so we can establish that bold information and hopefully have it for every known organism. Similarly related to that is this week's function of bioinformatics, where we use bioinformatics to study evolutionary relationships. So on tap this week is actually what we'd call it more specifically other than evolutionary relationships are taxonomic relationships. And this requires us to remember a little bit about what taxonomy is from Honors Bio. So, of course, what is taxonomy again? Taxonomy explores relationships between organisms. It's the attempt that we make to classify. Remember, it's our classification chapter, where we try to determine King Philip came over for good spaghetti or kingdom, phylum, class, order, family, genus, and species for all of the known organisms. It's where we assign each organism a genus and species name. Remember, genus is capitalized, species is not, both are italicized, and we call that binomial nomenclature. And taxonomy also lets us construct phylogenetic trees. You'll remember those trees, we had nodes and outgroups, and we drew them to determine relationships based on similarities and differences. When we did it in class, though, when we learned it during the school year, we used similarities and differences in structures, homologies, and analogies. We have a different way to do that now because of bioinformatics, where instead we construct a phylogenetic tree based on evolutionary relationship determined by similarities and differences among DNA sequences. This was an impossible task before DNA sequencing and genomics became as relatively inexpensive as it is now. I didn't say cheap, I said relatively inexpensive compared to what it used to be. Taxonomists would use a bioinformatics technique that we call multiple sequence alignments. So what you've done so far in your research is a single sequence alignment. You took your unknown sequence and compared it to the known sequence in the databases that exist. Here, what scientists would do is take a group of sequences from multiple different organisms and compare them to each other in that multiple sequence alignment. And it would look something like this. We would compare, so in this diagram we're comparing papaya, grape, tomato, and watermelon. So we would have 4 different sequences where we would have papaya compared to papaya, which you'll see the results of right here, being no differences. Of course, when you compare papaya to papaya, it should be identical. Comparing the grape to papaya, there's 1 difference. The tomato to papaya is 2 differences, and the watermelon to the papaya is 3 differences. So a taxonomist would look at this and determine the relationship between a papaya, a grape, a tomato, and a watermelon and say, well, with 3 differences, watermelon and papaya are more distant relatives than, say, tomato and papaya, because they have fewer things in common. So we can use this data on this particular chart, the number of nucleotide differences, we can use it to construct a phylogenetic tree. Hopefully you recognize these phylogenetic trees now that you've seen it. I understand it's been a while, we did classification a while ago, but this phylogenetic tree has nodes. Remember, that's what we called these are internal nodes, it's a branch. That internal node represents an inferred common ancestor. So we're saying here there is a common ancestor at this first node, right there, that first internal node, that relates the watermelon to the next closest common ancestor of the tomato and the grape and the papaya. And we're saying The end of each of these, this is called a branch. So the end of each branch is an external node, and that represents a single species. So every species is the end of a branch, and each species has its own branch. And we're saying here, because of where they are on the nodes being next to each other, that the grape and the papaya are most closely related compared to, say, the watermelon and the papaya. And each one of these black dots represents an internal node, which is the common ancestor. This diagram does not have an outgroup, but you guys will remember an outgroup in Honors Bio we said was a species that we include that doesn't contain any of the things that we're comparing. So I think one of the ones we did in class probably had like a sea lamprey on it, and everything else we were comparing was a cartilaginous fish. We put it there we call it rooting the tree. It's a very distantly related species, and it gives us a point of reference for evolutionary time, like how long ago those diverged from their common ancestor. Now remember, these black dots do not mean that a tomato evolved from a watermelon. It means that they possess a common ancestor. So be very careful to avoid that common pitfall of misinterpreting a phylogenetic tree. The point of this lesson for you this week is to perform a multiple species alignment and create a phylogenetic tree. You're going to use the organisms from last week's assignment. So you had a mystery organism. All of that data for your particular organism you can find on the Google site. It should all be in a zipped folder. I'll give you a couple pieces of information, advice / warnings. Really, I don't know how many of you this will be a problem for. I know that when I was using the multiple sequence alignment tools, I had some problems because I am using a Mac. So you have to make sure that your Java works. It worked fine for me in Safari. It did not work in Chrome for my MacBook. So just keep that in mind. It's a workable issue if you're, okay downloading some drivers and making sure that some of your software is up to date. But, just be aware it could be a problem. All right, good luck.